Our presentation today is on a new treatment that we have been experimenting with, which may have the capacity to target and eradicate cancer cells in the human body with minimal damage to healthy human cells. In our age of modern medical miracles, we seem to still be struggling to provide adequate end-of-life care for cancer patients and effective cancer treatments to avoid that end-of-life care to begin with. That is where our experiment is trying to correct the current state of things in a way that may revolutionize the landscape. Our objective is to examine the efficacy of colorectal, or of our treatment, which, as I said, was the revolutionary concept of introducing a virus that targets and eradicates cancer cells. I myself have great fascination with this topic. On the right, you can see my uncle, Kevin. He passed away this summer from colorectal cancer. And when I saw the opportunity to work on this, I was very excited. Again, we are looking to see if we can target cancer cells and ignore our healthy cells. This idea of using viruses has been in the collective mind of the scientific community for about 50 years, but only recently have we seen our technology, gene editing improve to a state where we can actually begin to examine and attempt these treatments. We chose the poliovirus for our experiment. First off, because it has a readily available vaccine, meaning we have a form of the virus that can be introduced to the body with minimal damage to healthy human cells. In our experiment, we introduced concentrations that never would be used in a typical virus vaccination, but again, it is less prone to attack and harm healthy human cells. It also happens to, we have also seen evidence that cancerous cells develop more poliovirus receptors than healthy human cells, which obviously means that our poliovirus can attach to cancer cells more effectively than it will against human cells, meaning will keep more human cells alive. Second off, it's fast replicating. We can have a short incubation period where we can moderate and adjust the treatment as we see results of its effect on that tumor. Finally, it very effectively induces cell death or apoptosis, much more effectively than other viruses we experimented with. Again, because cancer cells tend to have more poliovirus receptors. We also chose colorectal cancer because current treatments are ineffective, to say the least. In late stages, mortality rates are up at about 95%. It is very deadly. In some places, it's the leading cause of death. So the lack of effective treatment and high mortality rate makes this an excellent strain of cancer to target. If we could prove this treatment effective, we could eradicate it first and therefore save many lives. Our methods and way we went about examining the efficacy of this treatment will now be presented by my cohort, Calvin.
Perfect. Yeah. All right. So obviously we had to test the effect that the poliovirus would have on cancerous cells. So we did decide to experiment with this using 4 cancer cell lines as well as a normal cell line as our control. Right now, for simplicity's sake, we will be referring to each of these cell lines as cell lines A, B, C, and D for our cancer cell lines, and cell line F for our normal cell line. Our cell lines differed from each other, obviously, but only slightly. However, each of them did have a gene that did express a special protein that does interact with the poliovirus. Really, once the poliovirus did interact with these poliovirus receptors on these cells, apoptosis did ensue and the cell did die. And the way that we did use to kind of test this out, we did use 2 assays to analyze the presence of our cell lines and to give us a general idea of the amount of healthy cells within each of our cell lines. First off, we did use the endpoint titration assay, what we did was we allowed each cell line to grow for 24 hours at 37 degrees Celsius in a humidified environment. After the first 24 hours, we washed them and then incubated them for an hour with the poliovirus. After that hour was up, we then washed and stored them all for 7 days. After that, we then used the MTT assay. For this, we did treat each cell line with the poliovirus at different multiplicities of infections or varying dilutions of the virus. We then incubated them for an hour at room temperature, after which we did wash them and incubated them again for 2 intervals of 24 hours, recording our findings after each interval to kind of see what's been happening here. These assays, they did help us determine and measure the metabolic rate of our different cell lines and really measure any sort of changes that we could find in the metabolic rate after we introduced poliovirus to them. What we were looking for any sort of change in that metabolic rate, specifically lower metabolic rate, which does show that these cells were dying. After we treated each of these cell lines with the poliovirus and tested them with our assays, we did find that apoptosis did happen in each of our cell lines, really after we introduced the poliovirus to them. We were worried that the virus would spread to more than just the cancerous cell lines, however. So we did want to be able to test that. Obviously, the goal of our experiment was to find if poliovirus would be a viable treatment option for colorectal cancer. So obviously we had to test this. What we used a caste-based coliometric assay to test the spread of the poliovirus within each of the cell lines. For this, we treated each cell line at a multiplicity of infection of 8 with the poliovirus, and then incubated them overnight at 37 degrees Celsius. Afterwards, they were then washed and incubated for 24 hours. Really, these assays, they helped us indicate the effect that the poliovirus would have on each of our cell lines, on healthy cells and on cancerous cells, and really showed us if we were able to control or keep the poliovirus controllable to the areas that we want them to affect, such as cancer cells.
So I bet you're wondering what effect the poliovirus had on our cells. These are pictures of our cell lines through the eyes of a microscope. First of all, we are unable to tell, due to their limitations, which cells showed the most amount of cell death. What we can see, however, is the obvious and visible lack of cells after the time these cells were introduced to the vaccine. The rows, A and B, demonstrate the cell lines, whereas the columns represent the time after introduction to the virus vaccine. The first comment was directly after introduction, the second after 24 hours, and the third after 48 hours. Now, after later tests, we saw that these two cell lines, A and B, showed the greatest amount of cell death. In other words, they were the most successful. Cell lines C and D also showed great results of cell death. As can be seen, even after 24 hours, a significant lack of cells can be seen. This was only magnified after 48 hours. This final cell line is our line of healthy human cells. Though not seen perfectly, the amount of cell death was much more limited than in our cancerous lines. Though more tests are required, this shows an interesting find that at first glance, the virus vaccine doesn't seem to attack the healthy cell lines, at least not nearly as much. So the next question that needs to be answered is perhaps, how can we measure cell death? This graph here shows the effect that each cell line, marked by their individual colors, underwent as different concentrations of the virus vaccine were introduced to them. The x-axis, or MOI, is multiplicity of infection, which is basically just concentration of the vaccine. The y-axis demonstrates how much the cell line survived by percentage. As can be seen, as the concentration increased, so did cell death. The correlation between cell lines stayed pretty consistent, though our cell lines B showed the greatest amount of cell death. It should also be mentioned that as the viral dose increased, the amount of cells, or the amount of death in all cells, increased as well. Above the cancerous lines, our healthy human cell remained quite alive. In fact, at only one viral dose, roughly only 10 to 30% of cancerous lines remained, whereas more than 80% of the healthy lines survived. Now, this graph only shows the result of after 24 hours. Let's take a look after 48. Here, the results magnify what we have already seen. At one viral dose, there still shows a distinct difference between the cancerous and healthy cell lines. However, the healthy human cell line appears to have taken quite a hit. The results from our assays are an interesting find. The caspase assay, which measures the caspase, a product of apoptosis, shows how much cell death has occurred. The more caspase, the more death. This is measured by optical density, seen on the y-axis to the left. As more caspase is found, the assay deepens in color, thus meaning more cell death. Cell lines A and B, in red and blue on the left, show the greatest difference between the assay with the virus vaccine and the control without it.
So with these results, I have a couple interesting findings. We started this study with the intent to see if the poliovirus could effectively treat colorectal cancer cells. We knew that some lines had an overabundance of poliovirus receptor sites. And we can see from the graphs and our apoptotic activity that the cell lines A and B that have the most poliovirus receptors are most effective. But that's not the only thing that's unique about the poliovirus. On this page, I have two graphs. The first on the left is a cutout of the previous graph on the last slide. This shows us how many cells we were able to induce apoptosis in by using the poliovirus. And it is a pretty fair amount of our cultures. What's interesting is if we look at a previous study that was done a few years before we conducted ours, we can see that they used the viral lines E12, E15, E17, E26, and E29. They used some of the same cell lines we did, namely cell lines B and D are present in both our study and theirs. Cell line D was infected and apoptosis was induced in both studies by all of the viruses. But what's unique is in their study, the only virus that was able to even infect cell line B was E12. And apoptotic effects were fairly minimal, even with that infection. The poliovirus, on the other hand, was able not only to effectively infect the cells, but was able to also induce apoptosis, showing that it's a more effective treatment for these specific cell lines. But that comes with a catch. We don't know for sure if in vivo we'll see the same effects. We know that in isolation, the poliovirus is very effective at inducing apoptosis in cells. Once put into a body system, we may see different effects. So the next step in our research is to isolate a xenograft and then inject it into a host, either subcutaneously or through orthopedic options. Once we do this, we could administer doses of the poliovirus and observe the effects that it has on the tumor. If it's successful, we'll see the virus home onto those tumor cells and have a large amount of apoptosis in the tumor cells that we hope for, while minimizing the impact that it has on the healthy body cells. If we're successful with this, we may have a new option for cancer therapy that has a lesser effect on the whole body, meaning patients have better outcomes, but also we may have discovered a new treatment for multiple cancer lines. And this may become the basis and the springboard for future cancer treatments. Thank you.